Okay, Mike, I'll turn it back over to you to introduce our next speaker.
Super. Thank you, Hannah. I neglected to mention it, but our previous speaker, Dr. Sharang Liu, is over at the Brigham. Our next speaker is at Mass General, Christina Faherty, Dr. Christina Faherty. So we have great representation from the new MGB, Mass General Brigham Consortium, that incidentally also includes Mass Eye near where I am. Again, Christina's focus is on bacterial pathogens, especially Shigella. And she's trying to understand it to a degree that would allow the ultimate development of vaccines. Christina, tell us what's new with Shigella.
Sure. Thank you very much for the invitation to speak and the introduction. I'm just sharing my screen right now. Hang on one second. Okay, can everyone see that?
Yep.
Okay, great. So Shigella contributes to the global burden of diarrheal disease. And I'd like to start out with just noting that we are having success in this fight, and primarily due to things like clean water and sanitation, better nutrition, and the rotavirus vaccine. However, we still have a long way to go. Diarrheal disease is the 9th leading cause of death, at least pre-COVID, and accounts for millions of infections and deaths per year, especially in children under the age of 5 years. So the current challenges, especially with bacterial pathogens like Shigella and even E. coli and Salmonella, are malnutrition, antibiotic resistance, and a lack of effective vaccines. And this is despite significant amount of research that has occurred over the last 40 to 50 years, and actually understanding important mechanisms of disease. However, we're unable to really reach that successive therapeutic advantage, especially on the vaccine front. So just to provide a little bit of background about Shigella, it is a gram-negative bacterial pathogen, and it is very human adapted. So it was discovered in the early 1900s, and it has really co-evolved with humans. And that's something to kind of keep in mind as I present my work here today. There's 4 species of Shigella that are pretty much 90 to 95% homologous, and there's multiple serotypes that circulate the globe. For my talk today, we will be focusing on Shigella flexneri. So the pathogen causes watery diarrhea and bacillary dysentery. And to do that, it harbors a 220 kilobase virulence plasmid that is separate from the bacterial chromosome. And encoded on the plasmid are important proteins for infection, including invasion and virulence genes, as well as a type 3 secretion system. Shigella also induces the watery diarrhea by secreting enterotoxins, and then initiates that bloody phase of diarrhea by invading the colonic epithelium. And I would like to note, since Shigella flexneri does predominantly affect the global community, especially in developing countries, it is prevalent as well in industrialized nations in Europe and North America. And currently there's a Shigella flexneri outbreak in Vancouver, Canada. So as I've mentioned, we do understand mechanisms of infection for Shigella. And here is a model of what happens for invasion of the colonic epithelium. The bacteria can't invade on the top or the apical surface, so it needs to find and sort of access these antigen sampling M cells that are present in the epithelium that are normally there to sample the environment and to decide if we should be initiating an immune response. The M cells are actually few and far between in the colon, especially relative to the small intestine. Yet we know the bacteria are able to target these cells and exploit that antigen sampling uptake. Shigella first needs to, once it reaches the basolateral side, first needs to get around the innate immune cells, particularly the macrophages. So Shigella is able to sense once they are engulfed by macrophage and very rapidly induce cellular death. And then they are available to invade the enterocytes at the basolateral pole, and where they can then replicate and spread to cell, from cell to cell without having to re-enter the extracellular environment. So what we do know regarding tissue destruction is that that is mediated mostly by the neutrophils that are sent to the site of infection, and these neutrophils are actually what's responsible for the bloody diarrhea phenotype and the tissue destruction that we see as a result of infection. So as I've mentioned, especially classically, we've really appreciated that as little as 100 organisms are needed to cause infection through the fecal-oral transmission, and that the bacteria can invade in the colonic epithelium. Additionally, what we knew classically when you think about gene regulation and virulence regulation in the host was that the body temperature at 37 degrees Celsius was utilized by the bacteria to turn on the virulence plasmid. So in environmental temperatures, the virulence plasmid is turned off. However, once inside the host, the bacteria is able to sense that and turn on virulence expression. But besides those classic examples, there was limited information about what we really could understand as how well can the bacteria successfully transit the host, and how does the bacteria regulate virulence gene expression, especially pathogens like Shigella that do not want to waste energy expressing virulence genes too early and only like to express them when needed at the right time. So if you think about transit through the human gastrointestinal tract, there's many barriers that need to be overcome, especially low pH of the stomach for Shigella, surviving transit through the small intestine. And then once inside the colon environment, there's an anaerobic environment and needing to overcome things like mucus and the commensal population. So I became very interested in what happens during the small intestine, because that is where the bacteria spends the majority of its time prior to causing infection in the colon. It takes about 12 to 24 hours to transit through the small intestine. And what's interesting about the small intestine is that bile is pretty much localized to this area of the GI tract. It's only 5% enters the colon, so the majority is actually recycled back into circulation and deposited in the gallbladder. And another interesting aspect about bile is that it's bactericidal, so it's a really great innate host immune defense. However, pathogens like Shigella are known to survive it, and these mechanisms were not well appreciated, especially in the early 90s and 2000s. So the overall hypothesis for our lab is that Shigella resists bile and utilizes small intestinal conditions as a host localization signal to help optimize virulence. And today I really want to focus on the virulent aspect of some of the work we've performed. But first I wanted to introduce what we discovered when we exposed the bacteria to bile. So in the lab, we actually predominantly use a mixture of bile salts, since that is about 80% of the composition of bile. And what we can do is supplement bile salts into the laboratory growth media and then do our various investigations. So we found that Shigella can resist the physiological range of bile salts, which is about 0.2 to 2% weight by volume, depending on the person and diet. And we found a significant mechanism involved in this bile salts resistance was the induction of, sorry about that, the ACR-AB efflux pump, which is very much important for antibiotic resistance. It can expel at least 20 different classes of antibiotics outside the bacterial cell. So we found that these genes were induced in the presence of bile salts, and that an ACR-B mutant was unable to resist bile salts. And so this has important implications for antibiotic resistance. It's very much conserved amongst the enteric pathogens. They all utilize this pump to resist bile. And if you think about it for future therapeutic development, it's important because the sheer fact that enteric pathogens are exposed to bile at some point in the infection cycle is actually priming them for reduced antibiotic susceptibility or increased antibiotic resistance, depending on how you want to look at it. We also found that the bacteria in the presence of bile salts forms a biofilm. And this actually turned out to be an interesting story, because when we were utilizing different laboratory media like LB or tryptic soy broth and adding bile salts to that, we were noticing different phenotypes. So in tryptic soy broth, which has added glucose relative to LB, and then in the addition of bile salts, we were finding this biofilm formation happening. And so it turns out these both bile salts and glucose are needed to form a biofilm. And this is really important for the extracellular matrix that's produced by the bacteria when they're exposed in these conditions. So we really believe that we're starting to really understand transit through the small intestine with these mechanisms, and that bacteria will be exposed to bile salts and to glucose as food is being digested, and those simple sugars are being released by the digestion process. And this allows them to sort of transit the small intestine, and actually helps to explain why they possibly are not infecting in the small intestine and only in the colon. Now, we knew once we started detecting the biofilm formation that we'd have to understand a dispersion mechanism, because how else is the bacteria going to invade the colonic epithelium if they're inside a biofilm? So we began to investigate, and we found that the removal of the bile salt signal from the biofilm was enough to start a dispersion event. And so that actually mimics what happens as you transition from the small intestine into the colon, as I mentioned, where you only have about 5% of bile entering the colon, and a majority of bile is in fact being recycled. And then finally, we found extensive changes in gene expression through RNA sequencing analysis. So not only did we find changes to help resist bile, but also even things like suggesting an adaptation to the anaerobic environment. And then we saw significant induction of virulence factor expression. So our model at this point was, after the dispersion from the biofilm and resisting bile, that the bacteria are sort of entering the colon in this hypervirulent state, relative especially to growth in laboratory media, and that they're ready to cause infection. So we were kind of really interested in pursuing this hypervirulence, and some work done initially in the 1990s by Shelley Payne's group and Wendy Picking's group demonstrated that if you expose Shigella to bile salts, that actually induces the adherence and invasion phenotype. The invasion phenotype was worked out very well by the Picking's group, in which the bile exposure affected one of the invasion proteins, IPA-D. And then, so we knew that this phenotype was happening, and we were really interested in exploring the adherence aspect of it. So combined with the previous literature and what we were seeing on our RNA-seq data, we started to look at performing adherence assays with colonic HT29 cells. So if we grow the bacteria in subculture conditions with glucose and bile salts, we had a significant induction in adherence relative to bacteria grown in media alone. I would just like to note that we do standardize the bacteria to the same inoculum for these assays. And then also, we are only exposing the bacteria during the subculturing conditions. And so what we do is we grow the bacteria in these conditions, wash and resuspend in tissue culture media, then that sort of helps to mimic that small intestinal to colon transition, and then applying the bacteria to the cells. So there's no bile salts or glucose added to the actual epithelial cells. So we saw this really significant induction in adherence factor expression. And then, so that led us to the question, to adherence. So that led us to the question, does Shigella make adherence factors? And this is actually an interesting question when you look at the Shigella literature, because this consensus was that the bacteria do not express adherence factors. And that was primarily due to the fact that when you grow the bacteria in standard LB laboratory media, you do not see any adherence factors being expressed. The bacteria look completely bald. However, if you then compare that to E. coli under the same conditions, you'll see very much significant expression of the adherence factors. So coupled with these observations, and depending on which species of Shigella you're looking at, the genes are either not present to encode for the adherence factors, or they're present, but they're annotated as pseudogenes, either due to various mechanisms, which I'll get into in a minute. So we knew we had a little bit of a hurdle to overcome with this, especially with what's been established in the literature. So our first approach was to look for the adherence factors from both the biofilm assay and then also adherence to epithelial cells, and then go in and look at the genetics behind the expression. I just want to note that at this point, we started to move away from the traditional cell lines like the HT29 cells, and then started utilizing organoid cultures, which have become very popular in the past 5 years. They're a more physiological relevant model of different sites of tissues, especially for the gastrointestinal tract and for the lung. And so what's really an advantage to them as well is that you can get the expression of the different cellular types that are present in your organ of interest. And then also it's usually in a non-cancerous cell line. So at the center, with our collaboration with Dr. Stefania Sanger, she was able to utilize this modeling for her studies with celiac disease. And so basically, if you can isolate the crypt cells, the stem cells from the crypts of the intestinal epithelium, you can propagate them in culture and develop these spheroid or organoid cultures. And others in the literature, as well as Stefania, found that you can then trypsinize these cultures and seed them onto a transwell system. It's very similar to polarized TD4 cell lines and things like that. And so this actually was really helpful for us from a pathogenesis perspective, because we can then look at applying the bacteria directly to the apical surface. The problem with the spheroid cultures is the apical surface is on the inside of the spheroids. So absent of any type of micro-injection of bacteria into the cultures, this transwell system offered a really nice format in a multi-well format to do our analyses. And then when we do things like add differentiation reagents to the media, we can make sure we get expression of enterocytes, mucus-producing goblet cells, and those antigen sampling M cells that are really important for Shigella. So here we're just showing a top-down SEM view of the monolayer, where you can see that you get that same single cell that lines the intestinal tract. Anything fuzzy here are enterocytes expressing microvilli. These darker cells are cells that don't express microvilli, which are typically the M cells. If you do some transmission electron microscopy, we see really nice microvilli expression, as well as the nice tight junction and barrier formation that is important and an important function of this model for your intestine. And then, so we also looked at other cellular markers to make sure that we did in fact have expression of the M cells. And here we're showing we get very great mucus production from the goblet cells. So we really had a nice physiological model. We went ahead and characterized Shigella infection on the model, as well as some other enteropathogens like Salmonella and E. coli. And so we found, for the most part, especially when we utilized our subculturing conditions, we had great adherence to the enterocytes. As you can see here, again, the microvilli are on the top, and we saw nice adherence. And then where there were areas of cells that were not expressing microvilli, so most likely the M cells, we saw areas upon SEM analysis where it looked like the bacteria were starting to penetrate those cells. So then we went on and further characterized, and we found that the best invasion rates occurred when we had conditions that induced the M cells. And then comparing different samples along the gastrointestinal tract, especially relative to the small intestine and colon, we were getting the best invasion rates in the colon, indicating we had a nice model that replicated the tissue tropism of Shigella, and that also we can infect in a physiological relevant manner by exposing them to the apical surface, as opposed to directly exposing the bacteria to the basolateral surface. So this model is very helpful for our studies in the lab. And so back to the adherence story, when we looked at the bacteria grown in the biofilm or following adherence to the organoid model, after we subcultured the bacteria in bile salts and glucose, we could see great expression of adherence factors, or what looked like adherence factors to us, in both conditions. And so again, we are, if the bacteria are from the biofilm, the bile salts and glucose are present in the media. However, if there's adherence to the model, again, this is only during the subculture and not during the actual infection. And again, we can see the really nice adherence factor expression relative to growth in laboratory media alone. Again, we did need both the bile salts and the glucose signals, because if we grew the bacteria in LB plus glucose or just LB plus bile salts, we did not see the appearance of the structure. So we did need to have that combination. And so for the most part, we saw 3 types of structures, these thicker structures that you can see here, thinner structures, and then these protein aggregates that were very evident and disorganized. but still present. So we not only detected expression under these conditions in our laboratory strain of Shigella flexneri 2457T, but we also detected in other strains and clinical isolates of Shigella that we had in the lab. So we knew we weren't working with some fluke with our lab strain. And so just looking at the binding again, the three structures seemed evident for two different cell types, with the thin structures binding to the microvilli, and then the thicker structures and those protein aggregates binding to cells that lack the microvilli, most likely the M cells. So now we had a justification to genetically go in and look at the genes and expression of these factors. And a very brief crash course in adherence factors. Many genes are needed to actually express and secrete these factors onto the outer surface of bacteria. So for the most part, you have structural genes that are assembled on the outer surface that can be divided between major structural subunits and minor structural subunits. And then there's also the pores or the ushers that are needed for secretion. So you have the pore in the outer membrane, and then the chaperones that bring the individual subunits through the pore to allow for this assembly on the outer surface. So common types include type 1 Fimbriae and curli, especially in E. coli. Now, I know this slide's a little busy, but basically what it's showing is that for the most part in Shigella, the genes were intact and transcribed. So based on the appearance of the structures, we thought the thicker structures looked like long polar Fimbriae, the thinner structures looked like type 1 Fimbriae, and those protein aggregates looked like curli to us. And so we were able to utilize our RNA-seq data and map transcription to these operons, and then also confirm the expression of either the single genes or the operons by RT-PCR analysis. So for the most part, LPF, the genes looked very much intact for the expression. And to zoom in and focus a little bit on the type 1 Fimbriae, the reason type 1 Fimbriae was thought not to be expressed was because there was a transposon insertion in the usher or pore gene of FimD. But as you can see from our data that we do get significant expression, and this is about 90% of the gene relative to E. coli. So we were able to get expression of all the important components and segments of the operon, even though the operon itself is not co-transcribed together. And then for curli, we had the same problem with the transposon insertion, except it occurred for the structural gene CSGA. But again, we were getting about 90% expression of the gene, and then other expression of either the individual genes or the actual operon components. Also, the problem with curli was in the pore, there is a point mutation that leads to a stop codon. So we were able to confirm that, and that we would see expression up until the stop codon, but then not after that. So the annotations were correct. It's just that the annotations didn't mean that these genes were not being at least transcribed. And then one more experiment that we did to confirm at least transcription was to see if we can induce transcription. And so in the literature, glucose is a great signal to induce these major subunits. And so we did some qPCR analysis of bacteria grown in media with or without glucose, standardized the bacteria, of course, to the same amount. And so as you can see with our control, the glucose didn't affect expression of this gene. However, for all of these structural subunits and some homologs, we saw significant induction of the genes with glucose. So at this point, we were very confident that the genes were in fact being at least transcribed. And so we went ahead and made mutations in the major subunits of LPFA, FimA, and CSGA. And you can see we had significant reductions in both biofilm formation and adherence to the organoids. For the adherence to the organoids, especially FimA, was a significant reduction. We lost the structures upon TEM analysis as well. And then since we're mostly a bacterial genetics lab, we went through and complemented the mutations. We went ahead and made C-terminal histidine tagged fusions that would help us with some protein work. But basically, we wanted to see if these constructs would then restore function in the mutants and if the tag would interfere with function. So we did this for all three. I'll just show one example here with type 1 Fimbriae. The mutant again loses those thin structures, as you can see here. The complement very much nicely restored the structures, and we got nice expression on the outer surface of these structures, and you can really appreciate that we did in fact we're getting the right appearance as we suspected from earlier in the slides. And then most importantly, the construct with the tag was able to restore the biofilm phenotype. So just to kind of appreciate the appearance of the structures, looking at the complements, again, the long polar Fimbriae really appeared to be those thicker structures, the type 1 Fimbriae, those thinner structures. And then the curli, despite disorganization and a lack of assembly, especially relative to E. coli, they're there and it is functional. So we're really confident that we've identified 3 structures utilized by Shigella that are important for biofilm formation in the small intestinal conditions, but then also adherence and making that first contact with the surface of the epithelial cells. We went ahead and did confirm that the proteins, especially the tagged versions of the proteins, can be secreted and present on the outer surface of the bacteria. So utilizing the biofilm culture supernatants, we were able to detect the tagged protein secretion by mass spectrometry and immunoprecipitation analysis. So we have a model where we feel like we have 3 structures. Again, as I mentioned, we feel LPF is for the most part intact. And then type 1 Fimbriae, despite the truncation to the usher, we feel is being assembled and secreted appropriately. And then for curli, it is present, it is functional, however, it is disorganized. And that disorganization could be due to the fact that the pore is almost at a close to a 50% truncation, or that assembly is not happening due to the truncation on the major subunit of CSGA. So we're hoping to really dive into those mechanisms in the future. But with that, I hope to convince you that Shigella is in fact utilizing adherence factors and really has an interesting lifestyle to consider when you expose it to the in vivo environment or the small intestinal conditions. So with that, I'm happy to take any questions. I'd first like to thank members of my lab, both past and present, that have taken all of our work forward and have contributed with all their hard work. For this adherence project in particular, soon to be PhD Rachel Shannon, who is about to defend at UT Southwestern, really got us started, and Dr. Courtney Nickerson really took over as she transitioned to grad school. And then we had additional help with Alejandro for the adherence with the model. And we've had a lot of interns come through the lab that have helped us, both undergraduates and master's students and even high school students. And particularly Jessica Ding and Kelvin Lee, Harvard undergrad students, helped us with the complementation analyses, and Sneha Dakapathori and Benjamin Kuzbar that helped us with the RT-PCR analysis. Our collaborators at the center, Dr. Senger and Fasano, especially for the use of the model. And then for the RNA sequencing, Dr. Rasko and Sistrunk at the University of Maryland. Ms. Ann Tidsdale and Dr. Kumar have helped us with both the scanning and transmission electron microscopy. And then for the mass spec analysis, Dr. Auclair and De La Parra at Northeastern University. So with that, I will very much be happy to take any questions.
That was really terrific, Christina. And as we've announced before, if you have questions, you can use the Q&A function. I have just a couple. So Shigella, so as you know, I work on the gram-positive side of the fence. And so Shigella to me is sort of like a rogue band of E. coli's, right?
Yeah.
And so would you help me understand a little more clearly the ecology? You mentioned they're highly human adapted. Are there any other hosts for Shigella? Do they always cause disease in humans, or is there a high rate of asymptomatic carriage?
Right.
What do we know?
Yeah, so they cause disease in humans, and then they do infect higher primates, but that's it. So there's never been a really great animal model for Shigella, which has also hindered vaccine development and things like that. So it's pretty much restricted to humans and higher primates. And It depends on the species, but it does seem like it causes infections. And there are starting to be some questions if there's any sort of asymptomatic carriage of Shigella, especially in endemic regions like in Asia and Africa. So we're actually interested in pursuing that to see if, you know, what's going on with any sort of asymptomatic carriage for sure. But yeah, it's like I mentioned, it's very human adaptive, and that if you think about the human adaptation, you can really appreciate that it co-evolved with humans. And so it's going to utilize these signals to express virulence and prepare for infection, especially without wanting to waste energy once
Right.
inside the host. Yeah.
So unlike salmonella, it doesn't occur asymptomatically in turtles or anything like that.
No, not that we know of. So
Okay, no reservoirs. Let's see, we've got a question here from Dr. Chen. Fantastic research. I'm wondering if your lab has tested if any of those adherence factors play a role for colonization in a in vivo model, rabbit, mice, etc. Also, I'm curious to know, how does Shigella move between, I think, epithelial cells, but it says endothelial cells. Is that right? Or would it be epithelial?
Yeah, it would be epithelial. So we haven't, because of the human specificity, we've looked at the organoid models. There is a couple new rabbit and mouse models that are out there that look a little bit more promising to replicate human infection. So we would be interested in looking at adherence in that and help diving into the mechanisms. And then for the intracellular spread between epithelial cells, they utilize the actin polymerization like Listeria does, as Sarah mentioned during the lunch break. So that actually is really nice. It allows them to spread from cell to cell without having to re-enter the extracellular environment. So
Terrific. Well, that's the time we have. I'm still dying to do some Enterococcus and organoid experiments, so we got to put that together. But with that, Hannah, tell us about the art of Anna Maria Porras, and then we'll get back to questions for Christina and the others in our 45-minute post question and answer period. Thanks again, Christina.
Yeah, so I'm going to now queue up our final SciArt feature of the day.